the cloud. Hello and welcome, everyone. We're back discussing yet again chapter 12. There's so much in it. Chapter 12 of Stephen Grossberg's book, Conscious Mind, Resonant Brain. And we're focusing now on a couple of his basal ganglia models of sequencing, specifically sequencing and the balance between planned and reactive saccades. So it's a good time when as models get complicated to kind of sit back and say, well, what have we collected from all these models that we've looked at? Because I think this is what's really useful and can be reused in models that differ in lots of details, but you basically take some of these ideas and reapply them with new constraints. So automatic normalization or gain control is one of his earliest things that he stressed, and he got a lot of mileage out of this, particularly related to working memory kind of stuff and serial recall. And people seem to keep rediscovering the usefulness of normalization in the literature. So that keeps happening, but no one seems to use his equations for it. And that's a long story. And cooperative competitive dynamics. Again, people often talk about competition, but combining it with cooperation is somewhat rare. Positive feedback loops are less common in models, partly because they're so badly behaved. Top-down, bottom-up resonance, that's the version of the positive feedback that reinforces something where there's an agreement. Again, bread and butter of adaptive resonance theory. The 2x3 rule. So we didn't actually pause and look at this when we talked about art, but basically when you have a top-down and bottom-up resonance, if you look at a node that's receiving top-down and bottom-up, there's a third input that can come, which is exogenous arousal. And this sort of mechanism kind of recurs even if Steve doesn't call it the 2x3 rule, which is that you're basically making it so that a cell needs a certain combination of inputs to be above threshold. And it's quite useful for doing sort of conjunctive or and-like processing. And that comes up in some of these models too. Complement coding is another big one. So representing a thing and the sort of 1 minus that, the complement of that. And gated dipoles are kind of like a version of that has interesting temporal dynamics involving rebounds, and gets a lot of mileage out of that and becomes quite useful for resetting. And we have, as a general principle of hierarchical models, the hierarchical resolution of uncertainty. And so something that you can't tell whether it's an edge or not at a particular scale, if you stack up a bunch of those detectors, you can get a line. And then the consequence of doing this hierarchically may involve other problems that need to be solved. Sometimes it seems like you're solving problems that are internal to the model, but it's not a bad way to think as long as you keep looking at the data. Then there's habituative gating. It seems very sort of narrow, but it's really important in a competitive model to have a way for the winner to get sort of timed out so that something else can win. So it's a big part of art, and it shows up, versions of it will show up in competitive queuing too, and it's pretty useful. Basically, whenever you have positive feedback, you need something that can shut that off. And then particularly in the working memory stuff, in the queuing and sequencing stuff, you have primacy or recency gradients. Mostly we've been looking at primacy gradients, but you can get the same network to do the opposite, which is to kind of be in reverse temporal order depending on how the network is set up. So Steve sometimes uses the word spatializing, but that's a little confusing because it's not like Euclidean space or visual space, but just arranging something that had a temporal order as a parallel set of neural activities. And internal to the direct model and DIVA, all these motor control kind of models, is taking position maps, which could be some abstract state involving joint configurations and stimuli, and then mapping that to direction vectors. So rather than saying, I want to get to a particular place, you say, from here, I would like to push the system in this direction. And that can be learned through babbling, motor equivalent of babbling. I like this a lot, and it can be a little fussy to get working, but I think in future, it'll be interesting to kind of take insights from motor control and planning and try to think about higher cognition in those terms, rather than use these sort of more abstract ways of thinking about higher cognition. So, and spectral timing. So this we haven't actually talked about, and he devotes a chapter to it in the book, so we'll get to it. But basically, it's a way of appropriately timing an action by kind of assuming that you have a spectrum of possible delays caused by the end of a stimulus, and you can do reinforcement learning on them. So it's like sort of guessing all of the above possible delays and then you finding the one that's most useful. We'll get to that in a few weeks, I suppose. So basal ganglia, for a while during the era of cortical chauvinism, no one really talked about it, but it's become a lot more popular now. Subcortical areas generally are a lot more popular. Here are the main basal ganglia. So ganglia is plural of ganglion, and apparently in like a lot of nomenclatures for other systems, you don't use ganglion anymore for anything in the central nervous system, but because this is an old terminology, it's stuck. I think nuclei is what they use when it's in the central nervous system now. But so yeah, it's a little tricky to actually imagine what the 3D structure of this, because it's sort of like you have the thalamus as these two like ovoid structures, and you have these things that sort of curl over, around, sideways, and back around it. And I remember watching an anatomy video by some doctor from Pakistan, and he described the globus pallidus as part of this thing called the lenticular nucleus. And he said, imagine that that's like a piece of cheese, and the caudate and putamen is like a rat wrapped around a piece of cheese. Which I thought was quite a colorful metaphor. And if you like screw your eyes at it, you'll be like, oh, okay. But it's a weird one. But yeah, this shape makes it very tricky to understand where you are in the striatum, because that spectrum kind of matters.
The other one that really helped me, Yohan, when I was trying to learn neuroanatomy back in the day, is that the caudate sort of wraps around the ventricles. So,
right.
Which have, I mean, obviously they're also a weird shape, but if you think about them kind of like coming up here like that, and then the horn coming over in the front, you can kind of imagine the basal ganglia kind of wrapped around them in the hippocampus. Yeah. I just found that a useful thing as well.
Although I still find it difficult to imagine the ventricles.
Yeah, no doubt. You have to Google that image then.
Yeah, they never show us good diagrams of that. At least, you know, like modelers and people who don't do medicine. So I once remember I was like, that looks kind of like a Klingon bird of prey. but anyway, so yeah, looking at it in a more anatomical way is also handy. There's lots of circuit diagrams and models of various sorts. They're all more or less incorrect in some way or the other. This one on the left is from my PhD thesis. I did a model of basal ganglia based control of interval timing. And I used what is sort of the canonical picture with a few more recent additions. So there's these two pathways. The direct pathway, which starts with the D1 dopamine receptor expressing medium spinal projection neurons, which inhibit the GPI or SNR in the case of the ocular motor responses, and that inhibits the thalamus. So the net effect of the direct pathway is disinhibition of the thalamus, so it's sometimes called the GO pathway. Then you have the D2 expressing MSPNs that inhibit the GPE, which inhibits the GPI. So that's the indirect pathway, sometimes called the no-go pathway. And there's also the hyperdirect pathway through STN, which is excitatory and has a much more complicated kind of connectivity. And each picture I see of basal ganglia circuitry is sort of inconsistent in how they talk about the GPE and STN, but it does seem like the GPE projects back to the striatum. But I see very little what that does.
So there's the pathway, they describe this as the archipallidal pathway. And it has a really different kind of a characteristic to a lot of the other projections that you'll see in the basal ganglia, in that so it's ascending, it projects to multiple different locations. And if you actually look at the synapses, like Michael Bevan and John Paul Bolam have done some really cool work where they do juxtacellular labeling of these cells, and their synapses actually look like the glomerulus of a kidney, where they basically like enwrap local neuron cell bodies and like completely sort of like almost sort of smother them, such that they could almost sort of like, inhibit everything and anything they wanted. So it's a very different kind of a synapse, and I think it's remarkably poorly understood exactly what it's doing.
And Wikipedia has this unbelievable mess of a diagram, which just shows that schematic. I mean, a lot of people I know like to make their diagrams look like the actual thing, but it's just a mess. And the schematics are much easier to see, especially in this case. But yeah, so you'll typically see something like this, where you have the direct pathway to the output nuclei of the basal ganglia. The indirect pathway, there's another inhibitory hop. Hyperdirect goes to the STN. And a lot of these don't like talk about the other outputs of the basal ganglia. In fact, I recently was looking at a paper reviewing the motor system, which didn't even talk about the closing of the loop from thalamus back to frontal cortex, and was mainly focused on inhibition to mesencephalic locomotor region, PPTN, and other areas. So those are motor output areas. And for some reason, the papers that talk about these outputs don't seem to be overlap that much with papers that talk about the thalamic outputs. So it's a little bit hard to square these two types of outputs. Because on the one hand, people want to say, well, the basal ganglia is involved in action selection. And on the other hand, people are like, no, it's not really involved. It's something else. So there are some issues there. Another really cool aspect of corticostriatal structure is what my advisor Dan Bullock used to call the Haber spiral. It's not as well known as I feel like it should be, but there's basically a really interesting way in which the frontal cortical areas and the dopaminergic projections interact with the striatum. So the striatum is the gateway to the basal ganglia. And this is a slice through that has basically all of the parts of the striatum. The ventral striatum receives all these limbic inputs, so ventromedial prefrontal cortex, orbitofrontal cortex, those kinds of areas, as well as hippocampus and amygdala. And then you have OFC, which is also a fairly limbic area. And then you kind of have a spectrum. So it basically lines up pretty well with the cortical spectrum that you see of increasing laminar definition as you go along the cortex. So all of that projects onto the striatum. But as a lot of you will know, the whole cortex basically projects onto the striatum. So what I have not fully clarified, I dug into a little bit. One thing is the case is that this white bit that hasn't been labeled. So big chunks of the tail of the caudate, like the tail of that rat, receive visual input. And I was not able to trace in the 10 or 15 minutes I spent looking for where auditory input goes or somatosensory. But so, I mean, it seemed like when I saw this diagram of frontal and visual, it was like most of the caudate and putamen seem to be occupied, in which case the other senses are mixed in.
So Okihira Hikosaka has done probably some of the best work in this.
I was about to say, go read Hikosaka.
Yeah, so about the sensory projections.
Yeah, and the temporal lobe's projections to the tail of the caudate, and the kind of function that they might imbue. But the story, like, that kind of makes sense, I guess. And it's a kind of heuristic, right? But if you've got, hold on, so I can draw. If you imagine something like frontal cortex here, occipital, parietal, and temporal, is that you've got a kind of like a basal ganglia kind of circuit sitting in amongst all of those. And it just gets weird because the putamen has the strong connections with the motor areas, and then all the other areas kind of wrap around and have projections. And so the tail of the caudate really is kind of like the temporal lobe receiving area. The kind of prevailing story is that V1 and A1 don't have as strong projections. And then in humans and in non-human primates, I don't know enough about the rat or the mouse, but the tail of the caudate is really small, really thin. So Hikosaka's story about that is that you actually really don't need the basal ganglia to do its job in the temporal lobe, because the kind of inputs that get fed into that circuit are really, really quite stable. An item is an item and nothing else. It is this person's face and not that person's face, or it is edible or not edible. Whereas in the frontal cortex and the motor cortex, there's much more arbitration over exactly how do you reach out and grab that item, or should you go and get that berry or go for the other berry? And there's much more arbitration going on. but that's his take on it. I think it's quite a cool take, but that's basically the idea.
Good diagram because there's a big debate about it, like we mentioned briefly on Twitter also, that there's funneling and then there's segregation also, but then there's like, it's almost like a patchwork of Venn diagrams where there's overlap regions. So it's not a complete overlap zone either. So the way that the cortex interacts is it seems to create these patches, some of which are relatively dedicated to some corresponding cortical area, and some patches seem to be like blend areas. That's the impression I got, but I haven't seen a good diagram showing that. And I think
Susan Haber's probably done some of the best stuff there again. She had a really awesome J-Neuro paper. Haynes was the first author, where they tracked with viral tracing in a monkey a bunch of cortical projections down to the basal ganglia and showed that there were certain areas, they called them hotspots, particularly in the caudate. But then quite fascinatingly, the same structures that you would think would project into like the subthalamic nucleus, for example, if you're in the motor cortex and then dorsolateral prefrontal cortex, they'll project into different parts of the STN. Mm-hmm. Then all of a sudden, as you get down into orbitofrontal cortex, the projections actually hit the lateral hypothalamus rather than the STN. And so there's all this really interesting kind of deviations from the kind of like standard.
I may have actually seen that paper, but it was a while ago. So you'll often see a schematic diagram like this. Again, mainly focused on the frontal interactions. I've never seen a single diagram that has both the frontal and the sensory all in one map onto the striatum. So I'm hoping if anyone sees one, please send it to me. But yeah, so you have this cortical gradient, a striatal gradient, and a functional gradient. The functional gradient is a little bit up for grabs, and you don't hear about it that often in reviews of the basal ganglia. But the way I was taught it by my advisor, and which I think has a nice kind of, at least as a good hypothesis to begin with, is this idea that you have these limbic evaluative structures in the ventral striatum, which is more or less synonymous with the nucleus accumbens. It's not quite the same thing because there are some ventral parts of the caudate because of that wrapping. So you have to be careful about that. But the nucleus accumbens seems to be involved in very reward sensitive or outcome sensitive learning. And then you have this dorsomedial striatum, which is very connected with sort of more structured prefrontal inputs, which seems to be involved in action switching, and is particularly important during learning. And then as you sort of shift towards sensory motor association areas, you get these connections with dorsolateral, and eventually with motor, the corresponding striatal areas seem to be involved in habit learning. So there's this spectrum from basically reward sensitive learning to reward insensitive learning. And dorsomedial striatum seems to involve a kind of pivot between the two. So that's almost like a gate, which is that if something kind of passes the approval of that sort of loop, then it's sort of allowed to become a habit. And there's been a lot of conflicting data from the past 20 years to kind of mess up the whole nice, neat story people were telling up until the 90s. But I think this is a good place to start looking at the data. So with that, so the TELOS model is from 2004. So it was more or less operating based on the traditional kind of picture of the basal ganglia and how it interacts with the cortex, with a few sort of differences which make it potentially relevant to the debates about what the basal ganglia does. So the question, again, it's an excellent Grossberg question, framing a behavioral question. How does the brain prevent reactive movements from being triggered in situations where a more slowly occurring planned movement would be more adaptive? So it's almost something you feel subjectively. There's a prepotent response that kind of wants to get out there, and something stops it from happening. And how? So the model shows how quite a bit of data goes into this to show that you can prevent a reactive movement command from opening the gate before a planned movement command is ready to open it, and allow the reactive and planned commands to compete for dominance. And yet also allow reactive movement command to open the gate when no planned movement is being formed. So if you've got nothing better to do, go with the prepotent response. So it's called TELOS. The TELOS stands for telencephalic laminar objective selector. I think this is my favorite Grossbergian acronym because the word telos in Greek means goal or purpose. So it's a good acronym. So all of you must be well aware that monkey saccade tasks are like a whole cottage industry of neuroscience. It's just a vast number of, seemingly vast number of papers. Obviously, there are not that many monkey labs in the world, but there's a whole lot of emphasis placed on this sort of thing. Karthik also does this sort of thing. Nico does, or used to do this kind of thing too. And some fascinating data has come out of this. So the monkey's in head fixed, I believe, and has to saccade to different targets on a screen, and they get a reward delivered, typically juice or water delivered through the whole setup. And some of these tasks, nowadays, if you read about them, they can be quite complicated. They're rule based and like wait for a couple of things to happen and then move in the opposite direction to the target. So it can be quite elaborate. And the monkeys, they definitely overtrain, but they do things that humans would find quite difficult to do, even with a fair amount of training. So the specific tasks that they modeled were from this early Hikosaka paper. And so they have a fixation task where you just have to keep the eye on a fixation in the middle of the screen. A distractor comes on and you're supposed to just not look at it. There's a standard saccade task where you just look at a target as soon as it comes on. There's an overlap task, and I think that's the one where you have to hold on to the fixation while it's present and only saccade to the target when the fixation task, stimulus goes away. There's a gap task where, so it says here, what happens there? Yeah, the gap task imposes a delay between the offset of the fixation point on the onset of the target. The gap task target appears at a consistent location, and the subject learns to make an anticipatory pro-saccade to the target location during the gap between fixation. And last, you have a delay task where the subject has to remember the location because it's shown and then removed. And because the fixation stimulus stays on, you can't make a saccade until it's gone. So these are the tasks that they were looking at. So I like the way this paper is written, and I wish more modeling papers were written this way, but it kind of has to come together at the right point. So they had 14 computational hypotheses, which are pretty well justified by literature. A few of them not as many citations to back them up, but here they are. Forebrain circuits enable competitive limited capacity planning. And when they say forebrain, they're not committing to specifically cortex or striatum or something. It's basically the basal ganglia and the cortex are all part of the forebrain. And so they're saying that whole circuit is involved in limited capacity planning, meaning that there's not, there's sort of bandwidth limitations. The basal ganglia contextually gate expression of reactive behavior or plans. So they get a lot of mileage out of using this word gate, which sometimes makes it a little difficult to compare it with other people's proposals. But gating is different from sort of actively causing something to happen. It's a sort of permissive effect. It is not a driver, and it's not exactly a modulator, which is kind of a useful way to think, I think. Activation of the basal ganglia direct pathway can release plan execution. So it's not the same as executing. So it seems like maybe sophistry to talk about the difference between releasing a plan and engaging one. But the idea here is that the plan representation is separate from the release of the plan. So it's like, the flag that causes the cars to go in a race is not what causes the cars to go, but it releases them. Feed-forward inhibition in the striatum mediates competition for plan expression. So again, there are other ways to do this. Some people will try to use the medium spiny projection neurons just mutually inhibiting each other to have a local competition. They don't really use that. And there's reasonably good anatomical evidence that the mutual inhibition among medium spiny projection neurons is not good enough to implement the competition locally inside the striatum. Striatal activation requires convergent inputs from distributed plan representations. So, I mean, there's on one hand, there's this major fan-in, whether it's segregated or not, there has to be a fan-in. Even within segregated sectors, there's a fan-in. And the other important thing is there's a lot of evidence that the striatum acts a bit like a coincidence detector. It really, really likes spikes that are temporally well aligned. So, 10 spikes that come at random intervals versus 10 spikes that are very close to each other, it prefers the latter, which gives a lot of ideas for what the circuit is doing. The indirect pathway activation enables deferral of a chosen plan. So it's like the hold kind of signal. Thalamus-striatal feedback guides learning of indirect pathway stop responses. So this is really interesting, actually. It's a funny little technical issue, but they make use of the thalamic input back to the striatum, which many people rarely even talk about. But they have a good reason for it, to prevent sort of learned helplessness. But I think I have a bit about that later. Laminar maps enable gated interactions between planning and executive cells. So this is a version of things we've seen where we're using cortical maps to create conjunctive representations. There's a fair amount of complexity in this because you're dealing with different, spaces and different types of maps, but they get it to work. To have, so you have object representations and you have locations in head-centric coordinates and retinotopic coordinates, and you have to kind of make all these align. Small basal ganglia gating channels control large topographic zones in laminar target structures. So when they say laminar, they mean in the cortex. So that's a consequence of this funneling. So you have very, very many more cells in cortex funneling inwards to striatum. And there's an even bigger funneling from striatum to its outputs in globus pallidus. So you can ask, well, if the basal ganglia is mediating competition, what is it doing with respect to all that cortical real estate? Can it actually regulate all of those? And the answer is, they have an interesting answer, which is sort of, but not quite. So most of the selection is happening within cortex, meaning most of the direct competition between available plans. Selective gating of premotor zones enables fast sensory motor remapping. I don't know whether we get into that much, but sustained plan activity is a local cortical circuit property modulated by layer 6. So they have some very specific laminar hypotheses, which I haven't looked that closely into the physiology of the different layers. I can't, and they don't say anything much about layer 4. So whether it'll fit with what we've been thinking about on the side is another matter. But it basically, these are versions of what we've seen in simple working memory models from last week or a couple of weeks ago, because you have to maintain some kind of recurrent activation. In this case, related to the plan that you will execute when the fixation task stimulus goes away. Reinforcement signals guide learning of cortical and corticostriatal links. So there's nothing particularly complicated about that conceptually. Feedback from superior colliculus to FEF serves learning and performance functions. So yes, knowing that you have completed a saccade gives you a signal that you can use to reset and then move along. So that's one important feature. And cortical subthalamic signals enhance lockout of competing plans during plan execution. That's the hyperdirect pathway. So they make use of that too. Okay, so it's a fairly complicated model and not very easy to kind of go through it, even in the time I've given myself. So there's a lot happening, and obviously I'm not going to explain all of it, but there are two basal ganglia loops, a nigrothalamic and a nigrocollicular one. The nigrocollicular one is controlling the actual superior colliculus outputs via the SNR, whereas these others are more to do with internal working memory related representations. But we can go into this in a little bit more detail to say what.
Can I ask at this stage, Johan, how much, just like in your opinion, the kind of specificity of each of the projections and kind of just how crucial it would be if some of them were inaccurate? Just because we had a little bit of a chat on Twitter about this, that I think we've learned more about how the basal ganglia recipient thalamus projects back to the cortex, and right. I think that the kind of probably the biggest issue with some of the old segregated loop models is that they kind of assume this notion of like a cortical cell projecting down to the basal ganglia, which then disinhibits a thalamic region, which then goes right back to that same cortical area and sort of like opens up that cortical area to now have some influence. And that's a little harder to sort of square away with the notion now that these P and R thal regions typically are they're the matrix type more often than not. There are some exceptions, as always with biology, but to a first approximation, we should think of these more as the sort of diffuse type rather than the targeted core type.
Right.
And so my question is, like, how much that would affect the kinds of functions in the model if that was, too precise of a prediction.
I think, I mean, I haven't gone through the mathematical specifications in great detail. But from the qualitative effects, I can say that because most of the specificity in terms of action plans is happening in cortex, the thalamic feedback is basically at the level of sort of categories of action. And so we'll get to this, so the thalamus is not choosing between specific actions, especially specific actions that are represented in the same kind of zone. So they've left enough room in here so that you can kind of fix some of this. I'm not sure how much. So
do you think that's imbued by the dimensionality reduction, so that by the time things have made it through multiple different loops, steps of that reduction, there's like really no way to have precise feedback for action A17 versus A16?
Yes.
Rather, like A-ish things are good and B-ish things are bad.
Right. In fact, I have a very interesting hypothesis about two types of competition, which I'll get to in just a moment. But the wider point is actually really important for us to talk about, which is that the boxology and the general way of analyzing the behavior is more useful than some of the specific laminar hypotheses, because we can get working memory like phenomena with the resets that we like in other ways. Like, they amassed quite a bit of data to justify this, but it's not like there's a unique mapping between models, and as anybody knows, right? There's many ways you can get roughly the same kind of firing profile. The nice thing about this is they actually fit existing profiles of spike rasters, for instance. So, but it's not impossible that you could do this in some other way, and we could, make some hypotheses about the bursting layer 5 and really tease apart this 5A6 business, 5B6. Because I've noticed a lot of people have been citing my lab mate Basilis regarding the 5-6 stuff and how it projects to the thalamic reticular nucleus. And he himself says that we don't actually know how good that separation is. And for limbic areas, it's not actually that easy to separate 5 into multiple layers, or even 5 from 6 sometimes is quite tricky to separate. So when we're talking about corticothalamic loops related to limbic processes, emotion, motivation, etc., we have to actually really think slightly differently, because it's going to be a sort of degenerate version of a circuit that might look more sophisticated as you get closer to motor cortex. And that's something to keep in mind, that the motor cortex circuit should not be considered the canonical circuit. So it's another version of the implications of the structural model, and the fact that some cortices don't have layer 4, and some don't differentiate between 2 and 3 very easily. So all of that has to kind of be taken into account. But the basic idea of working memory, so the stuff I put in the beginning, the Grossbergian toolbox, I would still use as my initial approach to new data, which is, how do I get recurrent activity? How do I reset recurrent activity? And how do I get the recurrent activity to give me some sort of rank ordering? Because rank ordering is crucial for producing a sequence. And then the other stuff is basically prioritization, like use the basal ganglia to prioritize different things or prevent certain action plans from happening. So that fairly hand-wavy version of the model is a solid guide. And then for more specific things, you have to go point by point. So we can actually look at what they claim each area is doing. New data has been almost 20 years, so there's a lot of new stuff. But so the trained basal ganglia system, the whole thing, allows or prevents movements. The substantia nigra pars at the SNR tonically inhibits the superior colliculus and pauses briefly to allow the SC to generate a saccade. So again, it's permissive. It's not generating the saccade. It's allowing the saccade to happen if there happens to be a saccade-related command, which isn't in basal ganglia. If the saccadic gate is open before competition among stimuli resolves, the system may foveate the most contrastive target, or may attempt to foveate multiple targets simultaneously by averaging ambiguous SC activity. So there's several citations for this. I thought that's quite interesting, that if you fail to actually perform a competition, you may get a kind of pathological cooperation where you're averaging between things, where you have two targets and you head for the middle and you get neither. And that can actually happen. Yeah, so there's a bunch of citations about that. So the feed-forward striatal inhibitory interneurons keep the basal ganglia gate shut until competitive dynamics in PPC and FEF have a chance to select a unique saccade goal. So a lot of the deliberative aspects, so these kinds of tasks, the prepared saccade, the anticipatory planned saccade, they typically take longer. And why do they take long? The hypothesis here is that multiple cortical areas processing slightly different aspects of the task need to come to a consensus. And what that means is that they mutually reinforce each other, as we see in ARC-like models, and that is used for the coincidence detection in the striatum. So it fits very nicely. In fact, one kind of nice take-home message is that like viewing the cortex from the position of striatum is a very interesting thing to do, which is because they're like the kind of, they're receiving all these signals and they're sort of looking for the most coherent patterns among the cortical inputs coming in. So I like this striatum, the view from the striatum way of thinking. FEF and SC are individually sufficient to generate saccades, but in the normal animal, the SC is an important common pathway. So the SC is the main saccade generator, and there's sort of more infrastructure behind it to kind of regulate no saccade, basically, preventing it from happening. And the frontal ocular motor area add the ability to use, yes, okay. So what is the cortex giving you that the SC doesn't already have? Head-centered and other non-motor error coordinates, plus using working memory, conjunctions of features, and internal sequencing. So all of this addition is coming from the cortical real estate. So this is a sweep that gives you the basic idea of what's happening in this model. I call it a sort of tug of war. So basically, as I said, FEF and PPC are both providing signals, one about the object to be saccaded to and the other about the location, more or less. There's some subtlety to it, but they're both kind of summing in the striatum and both contacting the feed-forward inhibitory interneurons. So if they don't, so if you have, say, one command that doesn't kind of sync up with the other, then the feed-forward inhibition inside the striatum dominates, and you get no activation of the go pathway. Yeah, so pooled inhibition to each SPN can overwhelm a specific excitatory input. So you could think of the inhibitory interneurons in striatum as a kind of variable threshold that needs to be crossed to get anything out of the direct pathway. And then we have, so when competition resolves so that the movement plan is unambiguous, meaning that they're kind of converging on the same direct pathway, excitatory input to striatal SPNs eventually exceed striatal feed-forward inhibition. So it crosses the threshold that has been constructed on the fly. And that can produce a movement. Then, if the FEF plans a saccade goal that differs from the location of a strong visual stimulus, the competing frontal and parietal activities collectively drive striatal feed-forward inhibition to keep the BG gate shut until the conflict resolves. So here you have feed-forward inhibition from two cortical areas, but because they don't mutually reinforce each other and they're not actually inputting to the same striatal action plan, they're inconsistent. So they contribute to a pool of inhibition, but they don't contribute to a kind of summation of excitation. So that's the sort of tug of war here between the inhibitory process and the excitation coincidence detection. And that arrow there is kind of showing the between FEF and PPC is showing the communication that produces coherence. As the frontal cortex imposes its saccade goal on the parietal cortex, so this is a top-down imposition, the competition between saccade goals resolves. So there's competition happening inside frontal areas and inside PPC, and these cortical communications can help sort of resolve and push the competition to sort of settle on something consistent. And then an unambiguous saccade can be produced rather than trying to average between the two. So if you were to just sort of boost both of them, you would get inconsistent action plans, or two different go signals would be triggered. So avoiding mixed signals is a big part of this. Expression of two conflicting plans at the same time would result in incoherent behavior, and evidence for this includes the saccadic averaging that I mentioned. Some interesting, reaction time data related to this too. So incoherent behavior can be avoided if broad field-forward inhibition is complemented by a high threshold for activation of SPNs, which is widely observed in different ways. So as competition between plans resolves in favor of one winner, the cortical states become more coherent, in the sense that remaining highly active sites are mutually compatible representations that help to specify the winning plan. So this hypothesis computationally clarifies reports that convergent cortical afferents to distinct localized regions of the striatum originate from cortical areas that are themselves strongly interconnected. So this is where on Twitter I was thinking about that term from cat's cradle. There's this term, there's the karas and the vampire. So a karas is a group of people who are acting to, part of like a sort of like a cosmic team. They may not even know each other, but they were working towards the same goal. Whereas the vampire is like a fake karas. It's like not there. They call themselves by the same name. And I think his example in the book is Hoosiers, like people from Indiana. It's like a meaningless grouping. But I really like this. So it's like cortical areas can form karases on the fly to influence the medium spiny projection neurons. So we never talk about synchrony in Grossbergian neuroscience, but here's a nice place to use it, and I think again the view from the striatum gives us a solid way to ask why synchrony, or because it will improve your chances of making a neural karas.
Yeah, Kale's right. It's a granfalloon is the
Granfalloon, sorry, vampire is a two-person karas, right?
Wampeter is the object around which karasses and granfalloon or karasses form. So it's ice-9 in the in the novel, but I don't know. In this case, it'd be like what, the action.
Granfalloon is the fake karas. Yes.
Yeah.
That book is hilarious. Anyway, so yes. So now we have to look a little bit at that indirect pathway. In simple reaction time tasks, an oft-rewarded plan can be cognitively primed for execution, yet await a permissive event, in this case, the removal of the fixation light, that signals when it must be released. During the priming interval, excitation of SPNs in the direct pathway could inhibit the GPIO SNR and cause premature execution, unless such gate opening was stopped until occurrence of the permissive event. So this is the sort of thing that you often just don't think about when you don't think about the unfolding of the system in time. It's like a signal, once it's excited in cortex, there's nothing preventing it from going to wherever it has, wherever it goes. It just goes down its axons, right? So it's like, but I don't want that signal now. I want to wait. How do you deal with that? So the trainable go signal is one thing you can use. You can learn, how much to weight the direct pathway, and that's complemented by a trainable stop signal. So another example of opponent processing, which is a case of a version of complement coding. So you have a trainable stop signal, and so there's two distinct bases for non-execution: lack of input to the go pathway and strong input to the no-go pathway. So what's happening? The metaphor they use here for some reason is like bidding, like it's an auction. So to complement the BG parallel gating system, planning cells encode preparatory activities and send bids to the BG. And associated executive cells generate phasic output if and when their BG gate opens. So the terminology they use sits kind of in no man's land in this debate between action selection and response vigor. But the fact that it's about bids means that if you're bidding based on relative value, like some people assert that maybe the basal ganglia is doing some sort of cost-benefit analysis. And this bid idea fits nicely with that. There are some timing related issues about execution where it seems like in some cases, cortical areas that are involved in causing the execution of motor responses are already active before the GPI. So you have to like keep an eye on that sort of data, in which case what the meaning of the direct pathway is, it becomes quite tricky to understand. I suspect one thing that people agree on is that learning is really crucial. I mean, basal ganglia is really crucial for learning. So it may be that once something crosses a certain level of learning, you get quite a prepotent response coming out of the cortex. So yes, so here there's some very specific hypotheses about laminar processing, which I hadn't had time to sort of pick through them, but that's like a long process of going through all the available data from and sort of seeing where goes what. And this will take some time, but it's worth doing and we should do it. So yeah, a large portion of cortical fibers projecting to the striatum emerge from small pyramidal cells of layer 2, 3, and 3 to 5A. I think this is still true. I looked at some of that Gordon Shepherd stuff, and this paper also says more or less the same thing. And brainstem projecting neurons make up a very small proportion of the total population of cortical striatal cells. The vast majority are more superficially situated, smaller pyramidal neurons. This is important because you're not closing the loop back onto cells that execute, that go to spinal cord and brainstem. However, like I said, this is not taking into account the direct outputs of the basal ganglia to those brainstem pattern generators. And I am yet to see anyone talk about them all in one sort of in a modeling framework. So it's a bookmark to look at later.
How often did you find the whole IT direct, PT indirect thing in the literature? I found a lot of confusion about that when I've read.
Yeah, the problem is that I saw, Dan Bullock sent me on a recent paper, now it's a few years old, by Gordon Shepherd on this, which had some nice diagrams, and it seemed to kind of land as something that made sense to him. And I haven't been looking at what else other people have said. This isn't a paper from his group, but it says more or less the same thing. So I haven't done an exhaustive kind of search.
Yeah.
But, yeah, it's something, again, bookmarked that we have to look into. Yeah. Okay, so this
I think I was in the same case where I found like a talk by someone that fit with what I was thinking, and then I heard another talk where it was the exact opposite, and I was like, "Oh fuck, that's annoying."
Yeah,
but now I don't remember which way was which.
So it's tricky, yeah, and dealing with the issue of the different species, and also people looking at different cortical areas that are at different points along this continuum, can lead to a lot of confusion. Like I've seen review papers talking about the basal ganglia and not saying clearly whether they mean dorsomedial, dorsolateral, and that's not useful. If you want to enter into a debate about the basal ganglia, you should say which part of it you're talking about. So anyway, there's this issue, and they kind of point out that just for the pure anatomical reasons, the corticostriatal GPI projection is described as a funnel. There's a massive fan in. But at this point in 2004, the funnel usage had waned, and because it implied mixing. But now at that time there was more thought of segregation of channels. Right. So they hypothesize a one-to-many relationship between cells in GPI SNR and gateable cells in frontal cortex. This is what I was saying about so there's a fan in coming into striatum, then there's another fan in, and then basically you go to thalamus, and then there's a fan out going up to cortex. So how does that work in terms of specificity? So models must explain how the effect of gating can be highly selective and accurate, even though the number of cellular degrees of freedom in the GPI SNR is relatively small. They sort of gesture towards explaining that, but I think it's a big open question, how exactly all that works. It's such a huge reduction in dimensionality. And even Suzanne Haber talks about how...
Oh, sorry.
Go ahead, yeah.
I saw Mark Humphreys give a talk at a Gordon conference years ago on the substantia nigra pars compacta, which is like the VTA, right? But for the motor section. And he had this really cool computational work suggesting that there was like a low-dimensional subspace within that, within the neurons, that like there's a population level encoding that essentially still maintains the major kind of separation that you'd like in terms of the inputs coming into it so that they can be read out appropriately afterwards. So even though you had this huge collapse in the number of cells, you still were like representing the information, quote unquote, in a really efficient way. Yeah. And we just had him on our symposium listening, and he said he's finally writing the work up. So fingers crossed in the next little while, there should be something coming out that I remember being really cool, but I didn't understand at the time.
The impression I get. Yeah.
This really does seem to me like where synchrony and synchronization in the cortex really can play a role here. Like, and it's obvious that you need like synchronized activity in cortex to generate the activity in the basal ganglia. So
100%. So I also like this idea. Again, I kind of wish the paper had a diagram to show this clearly, but in between a single funnel and pure segregation is a really interesting nether region, which sounds more useful, which is a kind of mosaic where you have segregated, dedicated parts of that are devoted to cortex and overlap zones that are specifically sort of little laboratories to mix cortical representations, which occasionally may be useful. So that's what I'm going to be working with in my head from now on. So the term they use for the targets of all this is gateable cortical zones. They just made up that term. They're discrete. So for simplicity, the model defines 3 basal ganglia channels and 3 associated GCZs in FEF. But these discrete elements could be replaced by a continuum. Any 2 cells, this is what I was mentioning earlier, any 2 cells that represent different plans in the same gateable cortical zone are likely to compete via strong mutual intracortical inhibition, so that when the competition resolves and the gate opens for the entire GCZ, only the winning plan will execute. So it's like saying that department wins. I don't care who in the department is going to do the job, but one of them is going to. You guys sort it out among yourselves. That's basically what's happening here. If 2 plans pertinent to a given degree of freedom do not reside in the same GCZ, then they are less likely to compete via such strong mutual intracortical inhibition. So a large activation of one may not imply a small activation of the other. However, in this case, striatal competition can ensure that only one of the GCZs receives a strong go signal. So where you cannot rely on local cortical competition, then the hope is that you've stepped out of that gated cortical zone, gateable cortical zone, and then you can use that sort of long range competition. And what this reminded me of, for some reason, was the uncanny valley. So it's like certain sets of actions.
Were you watching Mark Zuckerberg's keynote from last week when you were thinking about this?
I've been avoiding that whole topic, but like if you think of the uncanny valley in terms of inhibition, right? There's like things that get so close that they're like no. But then there's a sweet spot. So I was thinking that plans that are for evolutionary reasons typically mutually exclusive are sort of packed together, but they're mutually exclusive by virtue of being so similar in some sense. So they're packed together to be inhibited strongly. And then outside of that, you have things that in some situations could be performed together, but it's still weird. And for that, you like in somewhere up over here, there's an additional, maybe another bump down, but that has to be handled by a separate mechanism. That was just a random association. Right. So what is the thalamus doing in all of this? So okay, yeah, learning necessary to generate a stop signal occurs during trials on which premature release of a movement leads to non-reward. In the model, such experiences can lead to learned activation of the indirect channel associated with the release movement, even though striatal activation in the indirect channel is not a normal part of releasing a movement. So this is quite an interesting little issue. It's like one of those issues that comes up as a consequence of modeling choices, but still it's an interesting question. How do you learn, presumably using some sort of Hebbian or associative learning, when the signal that you would like to happen next time was not active? So it's like, I can't associate the brake with anything if I didn't press the brake. And they have a solution to that. How does the brain identify which indirect pathway it should recruit in order to stop a given direct pathway? we propose that the feedback pathways from the thalamus to the striatum, here's where they use it, help solve this credit assignment problem by routing a specific teaching signal to the indirect channel associated with whatever direct channel has just activated. This is quite clever. I like this sort of thing. So it's like another use of like a kind of like an inference copy sort of thing, to so that the indirect pathway, despite not even being active, this copy comes in and allows learning to take place. So it's like silently the synapses are learning, and that's cool. And
How would it affect this circuit if, for example, the thalamostriatal pathway predominantly contacted the interneurons?
How would that work? Then the learning specificity would not work, unless the interneurons are specific, which I don't think is possible, or at least
they're definitely not.
Yeah.
So there's a lot of work to be done in this space, but there's some evidence that like parafascicular thalamus projects to the cholinergic interneurons of the basal ganglia. And so it has a big effect on them, but I don't know much about the functional space. I know that those pathways are really important for learning, but I don't know about it for credit assignment versus gating or something like that.
The TANs and the dopamine burst have a kind of push-pull relationship that kind of opens up a window for learning.
Yeah, that's how I was sort of thinking about it. A little bit like almost like a clutch. Like you put the clutch in a car, and all of a sudden you can move in lots of different ways. So the thalamus is saying, now's a good time for you to kind of reset a little bit, allow the system to move. Okay, now we're going to shut down again. Now you just do your thing. Oh, time to move again. But I mean, that's naive. I haven't done any modeling or anything like that. So
yeah, Dan has a nice review paper on specifically on the TANs and dopamine, which I was thinking of digging it.
Oh yeah, I'd love to read that. That'd be great.
Yeah, it's a nice little modeling based thing. So
I'd just like to point out that all of this dynamics comes out of the recurrent gated dipole work that Steve did with Nestor Schematic.
That was that the first spectral timing paper, or was there another one before that?
Schematic did do the first spectral timing, or at least the first simulated spectral timing paper. But the one I'm thinking of is the Reed circuit.
Okay.
I think is the first time these
ideas. That inference copy thing.
Yeah.
Nice. Yeah, it's a really interesting thing which you will never think about. I mean, you might think about it, but you'll rarely think about it if you're not actually implementing a model. Because it's like, I would like Hebbian learning, but I would like to learn not the thing that was just done, but the opposite. if you could learn that, it would be great. It's like if you had that kind of control. Well, the complexity nowadays is that people have found that the go and the direct and the indirect pathway are both simultaneously active in a lot of cases. So it maybe is solving a problem that doesn't need to be solved. Okay, so we can go look at a sweep of how a plan execute episode works. They start with priming a location, and that comes in this planning area in layer 6 first. So there's a 6 and 2, 3, 5A cooperation. And then you have the bottom-up sensory input also coming in. And from there you have some input also going to layer 5B, which will eventually cause the execution.
So this all looks like higher order thalamus or something.
Higher order thalamus. Yeah, exactly. So that issue I haven't quite resolved yet. And what Max said was like that some of this stuff, there isn't the targeted stuff, but they seem to be using mainly the higher order thalamus anyway. So I don't know whether they got lucky or they were very, very careful when they reviewed the literature.
Reading between the lines in a bunch of studies, probably.
Because that sort of thing, I don't know, maybe there was some data on this. So I mean, they were careful to look at Dumb and Strick and all of that stuff. So and Haber, so they were looking at all this, Yetarian and Pandya. Okay, so then you're priming layer 5B, but you're not actually releasing anything yet. So there's still no saccade. Meanwhile, a collateral from these planning cells opens up the direct pathway, which disinhibits thalamus. But layer 6 is also sending input to thalamus. And then thalamus, by virtue of being disinhibited, goes back up to layer 2, 3, 5A. And that, that produces and also to 5B. So there's a double, but yeah, this is again something you have to maybe look into. Provides that like the 2-3 thing. It allows the 5B pyramidal neurons, the big pyramidal neurons, to get enough input to release the saccade. And then through here you have like collaterals that help you eventually like shut off the command through the STM. Yes, and that this post saccade also does that. So after having completed the saccade, there's inhibition going to both the working memory representation of the plan and the 5B execution. So that's the basic sweep. It's quite complicated, but all the infrastructure is serving the purpose of being able to do this sort of behavior. So I can't imagine that any model that does this sort of thing will be less complicated, not by much anyway. And so they look at a lot of this stuff, which is worth looking into if you're interested in this specific set of behaviors. But yeah, it's hard to review all of it. So they generated 17 distinct types of activation dynamics. Classic Steve. Exhibited by electrophysiological recorded cell types, which also predicted the behavior of several additional cell types, which is good. Like you'd think, isn't this what people want from modelers? Apparently no, because it's hard, people just ignore it for the most part. But yeah, here's just an example of, in fact, which may be consistent with the simultaneous activation of the indirect pathway. So this is a delay task, so you have to withhold the response. So the indirect pathway, which is GPE, becomes active first. That's the data, and I think this is the model. GPE comes on first. And then this gap in this pause in GPI is what enables the thalamus to be disinhibited and allow and there's also a predicted transient that relates to the command.
What is that little down bump in GPI? Where's that coming from?
This one.
Yeah, 1.2.
Yeah, I don't know what that is. There's so many because there's so much, feedback. There must be some additional, either a drop in the excitation from one or the other. Oh, it might be ST. I'm not sure. I have to look at it. Yeah, that's a good question.
I mean, that to me looks like a straight up gated dipole rebound.
Right, but then there's that little additional thingy there, right? The other little thing, but yeah, I don't really know. So this is relates to this broad topic about, so when they eventually compare it to other models and they ask talk about these zones, and so consistent with data on distinctive sensitivities of frontal premotor areas, we have extended the module concept to denote groups of cells organized in gateable cortical zones, each mediating a distinctive sensory motor strategy. And those strategies are broad, and within them you can have competition inside cortex. But the difference, which is useful because this relates to this ongoing debate, is whereas previously the striatum has been suggested as the location of the experts in mixture of expert type models, in the present model of primate action control, the experts are cortical, indeed frontal, and the PG function is GCZ selector. So it's like, if you like, making the competition easier by whittling down the options is one way to think about it.
I mean, so is this like sort of like a, I don't know, like you can think of the cortex as maybe like being able to express lots of different contexts, and the basal ganglia being able to kind of, I don't know, map like a whole bunch of different things to the same thing.
Right. That in fact relates to a paper that Mac sent me also, this sort of dimensionality reduction with conjunction. So
yeah, Bargad, it's a really beautiful paper. It's super long. It's like
I read just the background part, and then I realized there was a model coming after it.
It's one of those lovely models that the name kind of gives you everything, though. It's the reinforcement driven dimensionality reduction model, so the RDDR. And so, I mean, the idea is something like, with a training signal and the fact that the cortex's kind of specificity has to be collapsed somehow before you get the feedback, you get some of the kind of key features of the basal ganglia's kind of involvement in that process. That's a really nice model. I mean, Hagai Bergman's like one of the world leaders in thinking about the basal ganglia. And I like that it embraces that kind of, it almost has like an element of the funnel as well as the loops as well, because whatever makes it through the funnel can win and like be the thing that constrains what goes on. It just isn't as precisely kind of a set of loops as the intuition from the original Alexander et al. Paper leads you to believe.
So yeah, I think that's the right approach, because it's like, I don't think anybody is going to say that the basal ganglia is not mediating any competition. The question is where, or that it's not involved in a competition. The question is what is competing, and it's not the specific action. And when I said that, I realized that even saying specific action is quite vague. What does it mean? We're not talking about muscle fibers. It's something at a much more abstract level, and even the cortical GCZ that wins, or that is allowed to be active or more active as a result of this thalamic disinhibition, will complete the job by picking something specific. So this next model is even more complicated, so I'm just going to briefly point to it. But basically, there's a model called list parse, and that combine that with the TELOS model we just saw, you get list TELOS, which basically takes up where the TELOS model left off, where there we have one planned saccade competing with the reactive saccade. Here you have a bunch of them. So the task is sort of like this, and they worked with Miller and Heisted on this, that you have a bunch of saccade candidate targets, and you may have to repeat going to one of them or the other. So a more complicated task. And I'd say it's a really nice model in terms of all the stuff that they've put together. They reproduced all the previous simulations from the Hikosaka task and added these new ones. And so the main infrastructure that was added was to deal with, so turn a CQ style model of item and order into a model of item, order, and rank. So the rank being the sort of number of times you have to execute the same target, which is quite a subtle piece of jugglery. And so the cortex, so they introduced supplementary I field, and they use, I think, one of the parietal areas to do counting, basically. They algorithmically implement that, but there's another Grossberg model to implement the counting with differential equations.
SPL.
Right, yeah.
And I just want to say, for the people who don't know, Matt won in terms of model difficulty level in our cohort.
Just looking at, I was reading, and when you've implemented a model like this, it's like I think even getting a basic CQ model with a good privacy gradient to work properly is itself a bit of a parameter hunt. But getting item, order, and rank to work is tour de force of Grossbergian modeling.
So I feel like I have to ask, what are the, what as far as Steve models go, right? There are basic elements, and they're all kind of combined in certain ways. I feel like there's you could put a diagram together that just combines the.
My first slide was basically that.
Oh, sorry.
I didn't make a diagram, but I basically did, because that's exactly what I thought when I was looking at this. I was like, well, no way I'm going to be able to explain how each box in this model works. But I know that the ingredients that I'm familiar with are there. Maintenance. So the big trick, right, is different kinds of maps, similar to the maps that we saw in the direct model and the DIVA model. Basically conjunctive. And that may be a big complaint that you might have about this type of thing, is that a lot of the heavy lifting in terms of converting from one space to another, from eye-centric to head-centric to whatever, or body-centric, is done kind of using a kind of lookup table-ish. which the brain has such a lot of real estate. Maybe that is how it's done, and you can get that to be set up using self-organizing maps without too much difficulty. But you may wonder, is there a simpler way to do this? Or I don't know. I don't have a simpler way to do it.
So as Eric Schwartz pointed out many times, the processing speed required here, the only algorithmic method that is available to the brain to meet its deadline to make the next saccade is a lookup table.
Okay, good. Okay. I vaguely
I heard
remember
Bjorn Merker make a similar argument that if you like cache out all of the delays across synapses, that to have reaction times on the level that we have, you just do not have enough time to do serial processing of like A to B to C to D. You have to have an inbuilt look up, and you go, oh, this is a good enough match for now. I'll just try that. And, you either respond or you don't.
That's a good point. Yeah, I didn't think about that because I came across this computation argument in the context of something Chomsky was saying. And I was wondering what basis they were using to say that search is very difficult, because if you're doing search using among primacy gradients, in the case of a Grossberg model, it doesn't take much longer unless everything is exactly the same or barely different in their primacy gradients. So,
well, I mean, even think of it in the context of, visual processing, like Anne Treisman or, Jeremy Wolf could get slopes for conjunction searches of items. Like they could say, oh, this is going to be a 3 millisecond per item slope. This is going to be a 7 millisecond per item slope. I mean, that's less than, the cell, an individual cell's recovery time in V1. I mean, it's like, it has to be done in parallel and has to be done through something like a lookup table with a little sauce at the end.
Yep.
Sorry, to go back to this like sort of like Grossberg element sort of like thing. To what extent do these things really compose? Like,
we could spend a long time on this, but one of the reasons, and I don't know whether I'm capable of it, one of the reasons I want to learn, and this is a bit of a tangent, apologies guys, but one of the reasons I want to learn functional programming is to do precisely what you just said. So it's been the dream, right? Like the dream for me is to make the Adobe Illustrator of modeling, where you drag and drop network elements of various types that have been, maybe you yourself made them earlier, and you want to have 5 competitive networks or 3 arcs, right? And it should not be difficult to compose them. And there may be some calibration issues, which itself is a nightmare. Which is like, how do I normalize the weights?
Well, with I feel like the composition issues with Steve type models is all in the parameters, right?
Yes.
It's not in the network, like the network structure, right? Like the network structures are,
all the units speak the same language, where the coding is by firing rate. And firing rate plus place code. So that's not an issue. The issue is when I have a unit like this that's receiving input from only one other area, and I have 5 areas, I have to normalize, but that normalization is not just average the 5 inputs. It has something else, because it will depend on the time scales of the different events. If something happens rarely, but I want to take it seriously, it may actually need a fairly high weight, may not need a high weight, you know what I mean? So there's all kinds of questions like that. So they're definitely, I would say the thing that makes me not want to try it is the normalization issue. Like, how does something that I've coded up change when I want 5 inputs coming in as opposed to 3, and so on. And also there's and they're not perfectly modular, in the sense that I may have a boxology of A, B, C, but then D, I might be aware that D actually reaches in and contacts the inhibitory neurons inside C. So
right.
That
you just muted yourself.
That's where things should presumably be like not composable, right? Like,
yeah, or at the very least, qualitative effects will be hard to know, because as you increase the connectivity between areas, you're bringing more and more areas, especially in cortex, that contact the local inhibitory neurons. What that does is hard to intuit. Why is it that every single cortical communication also contact local inhibitory neurons. There's almost nothing that's purely excitatory.
So
Yeah.
as far as I know, the only person to successfully execute that what you're describing is Rushi Bhatt, because
He went crazy as a result, I heard. I never met him. He left the year I joined.
well, he's a really bright guy. But he did manage to come up with a method of having like 8 different directions, in a laminar, visual network, all converge on the same interneurons and not have everything blow up in your face.
Right. Yeah. So, I mean, I know how I've been collecting these things. Just when I'm walking around, I'm like, how the hell would I automate this? And then I collect a few different types of normalization regime, right? There's the average firing, there's the peak firing, there's the min firing, and there's like postsynaptic considerations, presynaptic, like the cell has to be a certain amount active per unit hour, And so inadvertently, or maybe as a side effect, this becomes a model of homeostatic plasticity. Because if you can get a principled way to decide what the baseline weights are for a model to do anything, then you're kind of approaching the zone of homeostatic plasticity. But it's like several people's PhD thesis worth of work. So I don't know how to do it, but I'm keeping an eye on functional programming for this very reason. To like have bits of models that can be bolted together like Lego blocks, so that we can have more loops. Yeah. So again, this is just a whole lot of stuff that they did, but if you just look at the simulation results, you can kind of tell what's happening at this point. Having seen what you have onsets of different items, they get loaded up into a working memory, which is purely parallel, and then you read them off to produce, in this case, the saccades that are recalled based on what was just presented. Really beautiful results. And whenever, at least when I start thinking about modeling a behavioral kind of trial of some sort, I'm thinking in these terms, where the absolute value of the relative value of different activities is feeding into a competition.
I would also just like to point out that these simulations are inconsistent with the CAM gradients that were introduced in distributed ArtMap.
Oh, CAM, content addressable memory. Oh, inconsistent. Why?
Because the CAMs are going to squeeze the count cells.
I have to think about that. But yeah, the counting, I have to look at that model also. So,
well,
yeah, I mean, that was what I wanted to talk about with regard to these models.
Who've implemented distributed ArtMap fully.
Who implemented it?
I implemented it.
Yeah. You implemented it. Oh, nice. Okay. Okay. Yeah. Anyway, so yeah, that's all I had really. And the task, since some of us are interested in revisiting broad corticothalamic modeling, is to like settle on a working hypothesis for what the basal ganglia is doing. How different will it be from what is in these models? So I came across this from annual reviews, so saying that contrary to some predictions, the classic picture, PG output may not gate motor thalamic activity, which helps to explain some perplexing observations from lesion studies. I was like, but then I just found this. I didn't even finish reading it, but this paper is kind of splitting the difference and saying that the vigor model and the action. So the vigor model is the idea that the basal ganglia outputs aren't mediating a selection, but are merely regulating in an analog kind of way the vigor with which the already chosen action gets executed, which fits reasonably well with something that Paul Chisek has also been doing. So there's I think it won't be impossible to fit it into a version of what's already been done.
It seems like it can't be both sort of situation.
Yeah, and that's what they're saying. But the way they're saying it is slightly odd, because they're saying that the vigor model and action selection fit within a reinforcement learning framework, so that one relates a given state to the selection of an action and the other relates it to the vigor. So they're purely folding everything under associative learning. And I don't quite know how that will deal with these issues of like the temporal latency and stuff like that. So,
but that's, I mean, that's inconsistent with, for example, Mark Churchland's, findings that that you have a low dimensional embedding where the vigor of the action is encoded in the primary motor cortex.
Really? Oh God, I have to look at this now because I only recently started thinking about it. But I know that, for instance, just accumbens, there's a lot of good evidence for, behavioral vigor. But I was not aware that you could find a similar kind of response to dorsolateral striatum. So again, are people lumping the striatum when they shouldn't?
No. I mean, this is M1.
I was talking about the other, because like when people say that vigor is happening in the striatum, do they mean all of the striatum or, but M1. So, I mean, it makes sense to me, like, but.
That's just movement vigor, right?
Movement vigor, yeah.
Yeah. So there's like a hypothesis for Parkinson's disease, which is that instead of thinking about it as an inability to mediate the movements in your motor cortex, it's just that you don't have the motivation to move. You've lost the ability to kind of gate the kind of oomph to get going.
Yeah.
And it's just like, I mean, there are two different ways to think about the same thing, but I think that is more consistent with the kind of things that like Josh Burke is thinking about with this kind of motivational signal, that then if it drops, all of a sudden you get the kind of classical reward prediction error from the kind of Reed Montague kind of story, just for free, because that's the temporal derivative of a value, like a motivational signal is the reward prediction error.
Yeah, it's tricky, but yeah, I'll have to look into it, because I think that the thing that makes me concerned is if you get an execution signal before the GPI has done anything. That for me is the main concern, and I did see that mentioned in one of these papers. So other than that, you can, like, it's a tomato kind of thing, right?
Can you say that one more time? The temporal wait, no, sorry.
There's a paper, Rob,
the time derivative of motivation.
Okay, so let me unpack it. So yeah. Josh Burke is the senior author. Arif Hamid, Hamid, is the first author. They have a Nature Neuro paper from a couple of years ago, and they got rodents, I think they were mice, to do this task where they had to basically stick their nose into a port, and the value of the item could change over time. And they basically quantified, or they operationalized the animal's engagement as how likely they would be to keep their nose stuck in the port. And they basically found that the firing rate of the dopaminergic neurons would kind of scale over time with how motivated the animal was, how willing it was to keep its nose stuck in the port. And then they found that if they tracked the temporal derivative of that firing rate, it actually looked just like the reward prediction errors that you would typically see when people would either take away a stimulus that you're expecting or give you a big boost in a stimulus's value that you weren't expecting. And so the idea was that the kind of traditional dopamine as reward prediction error is just one way of thinking about it. It's like a phasic kind of really precise in time signal, but there's a much broader signal going on of generally telling the animal, this is a good place to be right now. Good things are coming. Stick around. This is, a good place for you to be. Exploit, versus nothing's going well. Time to throw in the towel. Nothing's working for you. Time to go explore somewhere else. So that's how they kind of frame it. And then Josh has a really lovely perspective paper. I think it's in Nature. That kind of talks about these kind of two different ways to think about the system, and how they're kind of two sides of the same coin. Like really like an effective way in which to kind of have an animal learn what is how to get the most out of their environment. I'm probably scrambling some part of that because I haven't read that paper for years.
There's a paper from not too long ago called "What Does Dopamine Mean?" also by him, right? So
yeah, I think that's the one where he kind of talks about those experiments and then he puts them in light of the traditional RPE story, which has been, really the kind of prevailing story in the whole literature for quite some time, 20 odd years, maybe longer.
Yeah, and I'm pretty sure that there's a little bit of selection bias because not all the dopamine responses are going to look exactly like RPEs.
Yeah, my other favorite dopamine paper is by Okihide Hikosaka, or review paper Bromberg Martin, and they talk about the fact that some dopamine neurons care about reward, some care about punishment, some care about salience, some don't have a particular kind of character that we can identify well in our current experiment. So it's a bit of a kind of horse blinders to think of dopamine equals reward, which then makes you even more grumpy that people in the sort of popular media say dopamine equals happy. And that's the whole of it.
I've been
Have you
fighting a crusade against that for a long time.
have you looked at Simon Hong's work with Hikosaka about how the precuneus gates what the dopamine neurons do?
No, but please send me a link because that sounds fantastic. I love everything I've read from Okihide.
Oh, sorry, not the precuneus, the lateral habenula.
Ah, yes, of course. Okay.
Oh, I have read habenula stuff. Yeah, that's awesome. So while we're on the topic of Hikosaka, another thing that he's done that I really love that you guys might get a kick out of is to kind of maybe present a slightly different hypothesis for why you get this change in more sort of ventral striatal inputs early in learning, and then much more kind of, I actually never remember what these are in rodents, but in humans it would be the accumbens is really involved early, then the caudate, and then the putamen. I think it's dorsolateral and dorsomedial in mice, but don't quote me on that. And a lot of people say, oh, you're going from goal-directed to habitual. But one of the things that Hikosada's hypothesis was that what you're actually doing is you're going from world-based to ego-based processing. So as soon as you come across something new, you've got no idea what it is. So you have to think of it with respect to all the things that are around it. You have to say, there's a new object. It's this far away from that other object. And, when I move it, it does this to that object. And everything is very much kind of in this sort of external frame of reference. But as you start to get better and better at understanding the degrees of freedom inherent within that object, think of like a weird thing, like a new knife you get in the kitchen, or like a lacrosse stick or something that has this weird balance to it. As soon as you start to be able to anticipate the consequences of it, the movements that you make with respect to it, you've now put it into an egocentric frame of reference. And so his thinking is that, well, maybe what's actually happening is that when you originally interact with this thing, you're using your frontal eye fields and your parietal lobe to kind of figure out what's going on. And they're just getting all this input down into the caudate. But over time, as you start to get used to the kind of anticipate all the different movements, all of a sudden the motor cortex can kind of handle it on its own. And so it's not so much about storing a habit, it's about learning how to deal with the thing that you're interacting with. And I just, it's not mutually exclusive, but I just really love that perspective, and it really opened my eyes to thinking about that system a lot.
This is all the Takakawa, Kawagoe, Hikosaka stuff in the early 2000s. It's a really beautiful set of papers. So basically, among other things, that really unusually for monkey literature, they waited to train the monkey until they had an isolated cell. So they actually, in the dopamine system, have a record of the entire training from like total naive to expert. And they had monkeys that developed different behaviors when they introduced spatial biases, which were reflected in the single cell responses in the dopamine system. It's a beautiful set of work.
That's awesome.
I'm going to stop the share recording. So.